The overall goal of this procedure is to determine an individual's oral detection threshold for oleic acid, or what may be more commonly termed fat taste threshold. This is accomplished by first preparing the solutions that will be used to assess oleic acid threshold. A base solution is initially prepared in a beaker using food-grade gum arabic, EDTA, and UHT milk. The second step is to prepare a series of 13 fatty acid solutions using paraffin, increasing concentrations of oleic acid, and the base milk solution. Next, a control milk solution is prepared in the same way, but without the addition of oleic acid. The final step is conducting the tasting session to obtain the participant's oral detection threshold for oleic acid using an ascending forced choice triangle procedure. Ultimately, the fat taste detection threshold method is used to show an individual's sensitivity to fatty acid and how that individual's sensitivity to fatty acids may be associated with fatty food consumption, BMI, or even development of obesity.
The main advantage of using the forced choice triangle ascending test to establish taste thresholds is that we find individuals experience less sensory fatigue, especially in comparison to other methods such as the up-down staircase procedure.
This method will provide advancements in the field of sensory nutritional science. For example, how the sense of taste may influence food consumption and ultimately diet-related disease.
Though this method can provide insight into fat taste, it can also be used to establish detection thresholds for other tastes such as sweet, salty, sour, bitter.